Good morning students, myself Chayanika Bordoloi, Assistant Professor, NEF College of Pharmacy. Today we are going to discuss about gram staining technique. So first of all, in introduction, gram staining is a method of differentiating bacterial species into two large groups, that is gram positive and gram negative. The gram staining is almost always the first step in identification of bacteria. It is a valuable diagnostic tool in both clinical and research field. So here all the bacteria we cannot identify with gram positive or gram negative. So there are sometimes gram variable and gram intermediate groups also available. So history, if we see, the method is named after its inventor, Danish scientist Hans Christian Gram, 1853 to 1938, who developed the technique while working with Karl Friedlander in the morgue of City Hospital in Berlin, 1884. In 1884, while examining lung tissue from patients who had died of pneumonia, Gram had discovered that certain stains were preferentially taken up and retained by bacterial cells. So Gram did not use a counter stain in his procedure. It was a few years later when one German pathologist, Karl Weger, 1845 to 1904, from Frankfurt, added a final step, staining with safranin. Gram himself never used the red counter stain in order to visualize the gram negative bacteria. So here this figure is showing the difference between gram positive and gram negative bacteria structure. So in gram positive bacteria there is basically two layers. One is cell membrane and another one is peptidoglycan layer, that is thick peptidoglycan layer. In gram negative there are three main basic layers. One is the outer membrane, one is periplasmic space, and another one is peptidoglycan layer, but that is thin peptidoglycan layer. So there are basic 4 steps in gram staining technique: applying a primary stain, that is crystal violet; secondly, using the grams iodine; thirdly, decolorizing with alcohol; and fourth is counter staining with safranin. So here basically this figure is explaining the same procedure with figure. And gram positive bacteria, after this process it will look violet color, and gram negative bacteria it will look with pink color. So in procedure we can say first we have to fix the smear culture, bacterial culture, with heat fix smear. Then we have to apply crystal violet for 1 minute. Then we have to add gram iodine, and then washes the with the alcohol. Then again we have to rinse the smear with safranin for 1 minute. Then wash off the safranin with water. Then we have to air dry the slide and observe under the oil immersion lenses. So this is the procedure of gram staining. That's all for today. Thank you.